Good morning, everybody. I hope you've had a very good party last night. For those of you who attended, and for those of you who haven't, perhaps you should buy tickets for next year. We're here for our annual ESHG-ASHG Building Bridges session, which is this year organized together with EMPAG. And I'm here joined on stage with my co-chairs, Heather Mefford, who is the chair of the program committee of the American Society of Human Genetics, and Sam Riedijk, who is the co-chair of EMPAG. So we have a very good leading team here, and we also have a fantastic lineup of speakers. But before I introduce them, I briefly wanted to say something about the Building Bridges session. So we started this, as you see from this slide, in 2014, the idea being that let's say in all the different countries in the world, we have a lot of challenges, and there are differences between countries. And between the American society and the European society, we started to build up these bridges. Now, of course, these bridges go beyond the European and the American society, and we include everybody around the world. But this is something that we at least organize together every year since 2014. And you can see here the different topics that have been debated in the last 4 years. So we started with incidental findings, with uncertainties in genomic medicine, genetic testing in children, really implementing clinical sequencing and what are the barriers in driving. And then already in 2016, we had a debate on germline genome editing in Barcelona at the ESHG. We continued about career path, actually, at the American Society. And what we have more, of course, in 2017, we discussed, because of the 50 years anniversary of the ESHG, ethical and legal discussions in the past, the present, and the future. And last year at the American Society, we actually took a topic which is very different on the Zika virus. So really showing the breadth of our building bridges. Now this year at the American Society, there will be a building bridges session about prenatal genetic testing. But here at the European Society, we'll come back to germline genome editing, because this is a field that is so much in development that we feel it's important to discuss it with you and with both societies. So I hope you're going to enjoy it. Here you see the lineup of speakers we have. So we've asked our speakers to give presentations between 10 and 15 minutes, so really sharp presentations discussing the different aspects that they've been asked to talk about related to germline genome editing. And then we'll ask all of our speakers to come on stage and have an interactive debate with you. So we've asked, as a first speaker, I will introduce Emmanuelle Charpentier, who is a professor and researcher in microbiology, and she's director at the Max Planck Institute in Berlin, and she's one of the inventors of the CRISPR-Cas technology. So we're really happy that she's here with us. We'll speak much more about her this afternoon when she's giving the Mendel lecture. But now she's going to discuss with us advances and challenges in CRISPR-Cas9. Emmanuelle, the floor is yours.
Thank you very much for the kind introduction, and thank you for organizing this debate that is, I think, extremely relevant in our days. I don't have slides, but I have some notes, so you will have to listen to me. And I'm sure that the other speakers will complement what I say, so I will start with really a general introduction. So as a matter of fact, you are aware of the fact that genome editing has an enormous potential, both for the understanding of biological principles, but also for the improvement of human, animal, and plant health. You are also aware that during the past 6 years, transformative technology has opened up unforeseen opportunities in the manipulation of genes and genomes, and this is the CRISPR-Cas9 gene editing technology. So this technology works as a programmable nuclease system that represents now the most recently developed molecular tool used in genetics, and has been harnessed as a powerful and versatile technology to modify precisely genes and genomes in their expression. So it really provides a lot of potential in the field of life sciences. So surely there were gene editing technologies existing prior to CRISPR-Cas9. So I'm sure you're aware of the zinc finger nucleases and the TALEN nucleases. The difference with CRISPR-Cas9 is that it has been acknowledged to be much more versatile, precise, and inexpensive to develop, tailor, and apply. At least the versatility is extremely strong, because you can modify genes in different ways, and you can also modify the expression of the genes with one technology. So it's really, it has really been developed as a very broad toolbox. So for these reasons, it has been rapidly and universally been adopted to engineer or correct mutations, modulate gene expression, and mark DNA in a wide variety of cell types and organisms in the 3 domains of life. So that's why it is also acknowledged that this technology has revolutionized life science research by the democratization of genome editing, but it is also recognized for its promising and potentially transformative applications. So I can speak about agriculture, that is maybe not the focus of this meeting on human genetics, but nevertheless. And so now plants and crops carrying new traits with the CRISPR-Cas9 technology can now be generated much faster and with higher precision than in the past. And this brings on a lot of advantages in the field of agriculture. One example may be the introduction of genes for resistance to infection by pathogens, and other maintaining genetic diversity in crops. Having said this, this also allows to reconsider the definition of genetically modified organisms for the agriculture field. So there are a number of discussions and debates in this regard, and specifically some regulations and decisions with regard to regulation that have to be taken by the European community. So in the field of medicine, genome editing with CRISPR-Cas9, as it was also for using the technologies talon nucleases and Zinfigure nucleases, but let's say really with CRISPR-Cas9 is generally considered to have a large potential in the treatment of congenital genetic diseases, but also in cancer and infectious diseases. So a very promising approach with regard to using CRISPR-Cas9 or other genome editing technologies in the field of cancer is really cancer immunotherapy. So the idea to use CRISPR-Cas9 to engineer CAR T-cell therapies that may have advantages over the current generation of autologous CAR T products. Having said this, there are a number of companies and also pharmaceutical industries working in this field. With regard to biotech companies, at least the company that I have co-founded with Rutger Novak and Stantford focuses on using CRISPR-Cas9 and developing the technology to treat certain human genetic disorders with first clinical trial applications that have been filed with regard to the possible treatment of genetic blood disorders such as beta thalassemia and sickle cell disease. But also, this company, like other companies, focus also on CAR T-cell therapies. There are other, for sure, genetic diseases that which we hope can be targeted by CRISPR-Cas9, and the future will tell us whether really indeed this is the case. Bringing also the differential aspect of using CRISPR-Cas9 technology for somatic cells and human germlines, bringing these intense ethical controversies around some of its applications. So surely the addition of the human germline with the CRISPR-Cas9 technology is highly controversial, because as you know, then the genetic alteration will be passed on to the following generations. But nevertheless, rapid progress in genome editing technology is likely to soon enable more precise DNA changes, also in germline cells, not only in human somatic cells, at a new scale, and opening the possibility to intervene in human inheritance. That's why it's important to really discuss this type of application. So some recent publications already show how far scientists have already come while ethical debates about the potential risks as well as the advantages of the technology and international regulations lag behind. So there is certainly need for more discussions and binding regulations on the global level to anticipate those potential applications. There are a number of laboratories that implement research in strict regulatory frameworks, I mean most of the laboratories, but we really have a responsibility, us as scientists, to make sure that appropriate safety and efficacy measures for any potential therapy that involve patients are taken, and that any use of the technology that is ethically questionable is prohibited. With regard to the risks of applying the CRISPR-Cas9 technology in the human germline, there is for sure, this leads to an understandable fear of the public regarding the outcome of a technology that is nevertheless quite young to understand in all its details. So that's why it's critical to go forward with the technology for research purposes while questioning applications with regard to human germlines. You have different also research that's been going on with regard to the application of CRISPR-Cas9 in human embryonic cells. And I would like to refer to an early publication and that was followed by the developmental biologist Katin Yakan at the Francis Crick Institute in London. So our group uses human embryonic cells, and others are also doing this way, embryonic cells that will be left over from in vitro fertilizations and otherwise discarded, to intentionally create mutations with the CRISPR-Cas9 technology with the aim to really perform research, that is to study the role of certain genes that are important to understand the early development of diseases, and for example, also the embryonic development. So these cells are not grown for more than 2 weeks and allow us to really understand critical mechanisms for certain diseases. Having said this, with regard to the human germline, we need surely a clear differentiation with regard to the regulations that are for the somatic cells and for the human germlines, and to really draw a clear line to also allow to the public to maybe understand really that researchers work under very strict regulations, but are not crossing certain barriers. There are discussions going on involving different national and international academies. You have for sure a number of bioethicists that have worked on the subject. However, it's always difficult to bring the policymakers, the physicians, the researchers, the developers, and also intellectuals asking themselves questions about genome editing together. So it's always a challenge, actually. Having said this, I believe that it is really, how do you say, important for the publicly funded research to really have clear statement as to be able to go further with the research on genome editing using CRISPR-Cas9 without, this is my own opinion, crossing the barrier to human germline editing. I am sure that you're going to enjoy the presentations of the next speakers, and that we can have a fruitful discussions. And I leave the stage to my colleagues, and I thank you for your attention.
So I'm pleased to welcome to the stage Kelly Ormond, who's a professor of genetics at Stanford University. She's also co-director of the genetic counseling program there and is very active in the genetic counseling community in the United States. She's played a very active role at the American Society of Human Genetics as well, and played a pivotal role in helping to craft the ESHG statement on human germline genome editing, and that's what she's going to speak to us about this morning. Kelly?
Thank you to Heather and all the organizers for having me. And my main task is to review the ESHG statement on human germline genome editing, and I do not have any conflicts of interest to declare. Our statement was published last August, so I think that many of you are probably familiar with it, but I do want to remind people that this was a large work group that was made up of half basic scientists who were working in this area and half individuals who come from a clinical background and/or a bioethics background. And we felt very strongly from the beginning that international buy-in to our position statement was important. So we ended up having 11 professional societies endorse this statement, and about half of them were involved in the development of the statement. And you can see that we have most continents represented through this. Like all of us, we started thinking about this in 2015 when these first papers came out from China suggesting that germline gene editing was feasible. And we began with the thought that these are not new issues. We've been talking about gene therapy for, who knows how long at this point. And we have lots of bioethics thoughts that we have been processing for many years. We didn't want to recreate the wheel, but we did feel like there weren't any data about current social views. And obviously, we continue to not have great sense of the safety and efficacy of gene editing, so that impacts our bioethical analysis. We very clearly wanted to strive for a proactive policy that balanced the needs of many of the stakeholders and that did not restrict progress, particularly around somatic gene editing. And one of the things that we did and published in our paper was we looked at all of the other statements that had come out by that point. And there was some degree of consistency, certainly, between the recommendations, and I think we'll hear shortly about the National Academy and many of the areas of overlap. But most of the groups were supporting basic research, and few felt that germline gene editing was ready to proceed clinically. When we approached our statement, we began to think first about two levels. What if this technology doesn't work, and what are the potential harms? And then secondly, what if it does work? What are the societal issues that we should be considering? I think certainly in the US and obviously in Europe and many other places, there are regulations in place to think about protecting patients from harm if this does happen clinically, but there's tremendous variation internationally about what those look like, and particularly around embryo research. And one of my colleagues who was a member of our work group, Rosie Azizi, and many others have published this map where you can actually just see, even if all you can see is the colors in the back of the room, that gives you a sense of the diversity around things like pre-implantation genetic diagnosis or human reproductive cloning or research cloning, and then gene therapies and stem cell research. We spent a lot of time focusing on the societal implications of this and tried to think about it from the standpoint of individuals, families, and society. And of course, we all jump immediately to the societal issues around eugenics, and particularly cost of these therapies and social justice and equitable access. So I'll just review our statement briefly. We broke the potential for germline genome treatments into the in vitro bench research aspects, clinical trials, and then clinical translation. And I think, not surprisingly, we felt that the last two, any sort of clinical translation, was not ready for prime time, and I think we continue to feel that way. There are a lot of gaps in our knowledge before we can ever get to that point, and I think we are beginning to make some progress in these areas, but there need to be clear criteria for how we measure off-target, how we decide what is acceptable and what is not, and really coming up with good thresholds on a number of levels. However, we did feel that there was no reason to hold back basic science research, and that if we did this in a way that there was appropriate consent and that donors were aware of what was going on and comfortable with it, that we could truly learn a lot from this, and I think we already have in the past several years. Importantly, our group felt very strongly about mentioning public funding in our statement. And in the United States, there is an act that essentially prohibits federal funds to be appropriated for embryo research. And this has been in place for several decades. No US president has been able to override this. It continues to be in place. But what it means is that embryo research essentially has to have private funding. And that really does change the way this is regulated. And so our group, while we did not think that we had any chance of changing this in Congress, and I still don't, we felt strongly enough about it that we really did want to put it in our statement. Okay. Our final point was that we laid out 4 general areas, and this is very global, and I think we continue to be at this global point, where we believe we need to have more work before this could ever clinically move forward, if it does at all. The first is a compelling medical rationale for the particular conditions where this would happen. The second is the evidence base, which is obviously growing, but still not adequate. The third is a clear ethical justification, and I know that'll be something that my colleague Alta speaks about in a few minutes. And then the fourth is stakeholder input and transparency in process. So I want to just take a minute to talk about some of the stakeholder work that has been going on in the United States over the past couple of years. And this has really grown out of many of my colleagues on the work group and obviously some other individuals that I know about. And yesterday, Heidi Howard spoke briefly about the fact that she has a European grant to begin to look at many of these issues as well. So my colleague Kiran Musunuru has surveyed cardiology researchers. I'll show you a few slides of a survey on genetics professionals that we did and presented at ASHG last year. And then in terms of individuals and families who are affected by genetic conditions, there's a paper that's in press right now at Genetics in Medicine surveying families with Down syndrome. And then we have a plan to survey families with trisomy 13 and 18, as well as do interviews on all of those families with aneuploidies to see their thoughts about this. And then I'll present some information that a student of mine just finished interviewing patients with retinitis pigmentosa and Leber congenital amaurosis, just to give you a flavor for what we're hearing at this point. We studied qualitatively through interviews patients with retinitis pigmentosa, which can have age of onset anywhere from childhood to adulthood, and then Leber congenital amaurosis, which typically has an onset in infancy. And what we found was that views towards both somatic and germline gene editing were tied in part to your age of onset of this condition, but also importantly, how you viewed this condition in terms of its impact on your life and your sense of identity around blindness. And I think this is something that the disability studies literature predicted we might see in some conditions. So that was important, and these are just a couple of quotes about how people felt towards the concept of gene editing. So I'll read them just briefly. I can't think of any area where my visual impairment doesn't impact my life, and I will use that word negatively. That doesn't mean coping isn't in place, but boy, life sure would be a little easier. Okay, a lot easier. And I think that really resonates with some of the comments that arose from patient and family stakeholders at the National Academy meeting as well. This desire from patients, if they see conditions to be a medical issue that they would like treated or cured, they want these therapies available. And then on the other hand, this individual that says, I understand that to many, visual impairment is a very negative thing, something to be fixed. But as someone who lives with this condition, I never really thought of it like that. It was more just part of me. I can see why such technology was developed, but I never thought to myself, oh, I'd be so much more happy if only I could see clearly. And I think we're going to begin to hear from stakeholders that they're going to weigh the pros and cons and how much this may actually impact their medical condition and how much risk they're willing to take to get that done. These next couple slides I'll show you briefly are from a survey that we did where we emailed individuals and professional societies across the world. We had 500 respondents, and again, this was presented last year at ASHG but has not been published. You'll see that as genetics professionals, we had quite high support for somatic gene editing therapies. That's these top numbers, all above 85%, and then that decreased as you got into different scenarios around germline gene editing, depending on whether or not it included a viable or non-viable human embryo. When we looked at views towards somatic gene editing in therapeutic ways and then enhancement ways, this kind of salmon color here is strong support, and this is support. So you can see that for therapeutic somatic gene editing, most genetics professionals were quite supportive. and that decreased as we laid out different scenarios around enhancement. And that all of those views were a little bit reduced in terms of support for germline gene editing. And again, more for therapeutic treatments than enhancement. And then finally, when we asked genetics professionals if there were any features of the disease that impacted their degree of support, so things like penetrance, prevalence, age of onset, impact on lifespan, and degree of impairment due to the condition, we did actually see differences across all of those as well. So it really does reinforce the need to get more stakeholder data. And so I'll end there and look forward to hearing more from our additional presenters. Thank you very much for the opportunity to present.
Well, good morning. It's my pleasure to introduce to you Dr. Luigi Naldini. He's from Milan here, and he's a professor in cell and tissue biology and gene and cell therapy, and he will share with us his thoughts on this subject. Thank you.
Thank you. I'll just get out of this presentation here, though. Oops. It's not going our way.
Sorry?
Oh, okay. That's good, thanks. Thank you. So good morning, everybody, and I have the privilege to report on the work of the Human Genome Editing Committee of the National Academy of Science, together with the Academy of Medicine of the US. This study was actually involved a large number of different people who are represented here, actually including one of the two co-chair, Professor Alta, Charo, who will speak after me, and she will be back better suited actually to report here, but you will hear from her as well. And there were scientists, bioethicists, social scientists, representative from different stakeholder area, from the business community, patient association, and also different background in terms of religion and views. The way of working of the committee was to hold 5 meetings throughout a span of almost something more than a year. 4 of these meetings were held in the US and 1 in Europe, in Paris. And there was both an open session in which we invited different experts from different areas also to get a view, including like the industry, public engagement expert, and so on. And then we had closed session to work on those input as well on the existing relevant literature to come up with an analysis and eventually recommendation on how to potentially regulate the progress of gene editing. Now, in terms of the way the methodology taken was trying to reach out to as wide an audience as possible, although, of course, the specific mandate for the recommendation was to the US, as well as the UK and China. They were also sponsoring the study, was actually to found to use as a foundation of our reasoning international convention which deal with protection of human rights, as well as the proper conduct of clinical trials, to inspire the overarching principle of what could be our recommendation, with the view that this principle would essentially be potentially embraced by most countries and essentially built into the specific regulation that they would draw from them. Now, these are well-known principles in terms of ethical conduct of clinical research, starting from promotion of well-being, which help us to identify as the key goal of genome editing the treatment of severe conditions like disease or disability, and transparency in terms of full disclosure of the process of the research, and also engagement of public input on that process. Due care in terms of looking for careful stepwise progress of research, as knowledge is built and proper information is gathered on this technology. Responsible science goes as well in terms of the type of mechanism and integrity and oversight that we would envision to be built into any process of testing this technology. Respect for person, and that has to do, of course, with the dignity of person who would be participant to the trial, with proper respect of the personal choice and rights, fairness in terms of equal distribution of the burden of any study, as well as access eventually to the benefit coming from this technology, and transnational cooperation which acknowledge the diversity that different countries may have in regulating this technology, where this diversity may provide value, which have to be respected and valorized, as well at the same time, however, to ensure for proper exchange and freedom of scientists across different countries to work on this. Now, based on these premises, we have analyzed the key area where genome editing is being applied, starting from research, which is essentially where we all agree with the wonderful tools that this technology provides, going into somatic gene therapy, where you heard Emmanuelle Charpentier mentioning some of the key potential advances that genome editing can give us in terms of treating severe conditions, editing T-cell ex vivo for immunotherapy, blood cell as well, or directly in vivo editing muscle or brain or liver for different disease. Now, in general, somatic gene therapy is of course, effectively regulated by existing framework, starting from institutional review boards, which address the ethical aspect of the participation of subjects in trials. In some countries, like the US, there may be an advisory national committee which look into novel technology when they have to enter clinical trial and essentially advise the authority who has the responsibility to allow starting of the trial. In EU, this would be national authority for phase 1-2, which then, however, would go to the European Medicinal Agency for any process toward registration of new trial. Now, this framework may provide a sufficient level of regulation for somatic therapy, provided that we establish some of where the key goal of this approach will be. And the committee thought that at the current state of development, it would be advisable to limit clinical trial of genome editing at the somatic level to treatment or prevention of severe disease or disability. And this also would provide a proper framework of assessing risk-benefit as for any other medical treatment, essentially, as a way to allow proper development of the technology. Essentially, this may help us to deal with the complex area of treatment versus enhancement, where enhancement would mean making changes which go beyond the ordinary human capacity and essentially or outside of treatment or prevention of disease. Now, enhancement raises a significant public concern from a number of sources, but at the same time, enhancement is currently part of our societal life at very many levels, and there is a continuous, actually, growing changes in attitude toward using sometimes even what were medical treatment for enhancement. And you can think about editing where you could address, for instance, height or muscle strength, both to improve, to treat a disease like nanism or like dwarfism or, for instance, muscle dystrophy on the one hand, but at the same time, the same technology may actually augment what could be height or strength in general. So based on this, it's difficult to at the moment, consider that this type of enhancing approach would be suitable given the unknown and the potential risk associated with early testing of genome editing. So we felt that it could be advisable to not allow enhancement type of study to proceed at this time, even at the somatic level. And essentially, unless there is a more supportive and widespread consensus of such move. Now, heritable genome editing is really the key challenge that took most of our time in the second part of the year in the discussion. As you hear, this is really the focus of this debate. As mentioned, this is a realistic possibility. We are not talking anymore of transgenesis, which by definition, being imprecise, was never considered a suitable approach for modifying the human germline, although it has been used, of course, in animal and in crops. Genome editing, because of its precision, may indeed allow individuals to have genetically related children without passing on disease-associated alleles. There's a lot of potential alternative to that goal, which, of course, this is not a treatment. This is a prevention of transmission. So it's a different, of course, approach. We should not, I think, confuse the two areas here. Prenatal, pre-implantation genetic diagnosis, prenatal, and abortion are alternative, which many may feel as well not to be entirely ethically acceptable. So there may be limited cases where germline editing would be the only alternative to prevent this passing on to the next generation your allele. Situation where you have like homozygotes with a dominant disease or compound heterozygotes, or situation where there will be a very scarce number of embryo generated for IVF, which may make a selection at PGD difficult. So there may be area, although few at the moment as far as we envision, where this could be doable. Now, germline editing has been achieved in animals, and although there's still several issues in terms of efficacy and safety, which obviously prevent its application as of now to humans, one can conceive that these technical hurdles will be addressed possibly in a very short time. So essentially, you can look also how you would do that. And you could think of editing the zygote of the early embryo, although this may be unlikely to be the main path because of the issue of potential mosaicism and also the lack of validation of edits. There will be no way to know whether it has been proper or not. On the other hand, more likely, you can think about editing a germline cell, like spermatogonial stem cell, which can be harvested from the testis, grown ex vivo, and modified, and potentially where the proper edit may be selected, and then the edited cell may be reimplanted in the testis for proper generation of sperm, or in vitro differentiated to generate sperm for in vitro fertilization. And this has been shown, again, in animal model to work. Potentially, we also think about using germ cell, which can be either derived from the proper embryo or, more likely in the future, from adult cell through the process of induced pluripotency, which may generate as well primordial germ cell eventually to be differentiated into ovogonian and oocyte. So it's possible to get to, in fact, germ cell which have been selected for proper edit. So this is a realistic possibility, although not immediately applicable to human. So given that, of course, we have to address what the concern would be there before this technology will be applied to humans. And of course, the key aspect is that the genetic changes will be inherited by the next generation who could never consent to that intervention. There will be a multi-generational risk, but also possible benefits from that treatment because you would avoid transmission of a disease or of a severe condition. Now, the lack of consent is an obviously important issue, possibly intractable in ethical terms because the future child is not there at all. It's not even existing at this time. But at the same time, we should not forget that we are treating, for instance, in somatic gene therapy, children in the first few years of life where the consent is coming from parents and is being effectively addressed. The need for long-term and possibly lifelong follow-up, again, would be an important hurdle for the people born, at least the first people born with a genetic edit. But the same, this is also what we are currently doing in somatic gene therapy. And this has not been an insurmountable hurdle. There are, of course, more philosophical issues in terms of what level of intervention in nature is deemed acceptable, and how does this go with our growing knowledge of the genome and its function. And of course, this is an area where everybody can take a different view. Importantly, though, germline editing to prevent the transmission of disability may impact the acceptance in general of children with disability, may build pressure against or towards elimination of those disabilities rather than supporting them. It may overall undermine human dignity, as you can look at somehow the production of a designer baby according to certain requirements. But here again, our society has already dealt to a certain extent with that, where children sometimes are conceived to provide a matching donor for another earlier, older child who has developed a disease like leukemia, who would not have a suitable donor. Parents may decide to conceive another baby who will be selected for properly matching donor haplotype. So this is essentially that there's room where society may even go towards a designer baby, which we more suitably call savior child. And of course, eventually, this means that you may unearth the spectrum of eugenics in the long run. So this concern should be addressed in terms of balancing individual benefits, like the passive versus active right to parenthood, where, of course, the strong call for having children of a genetic relatedness has to be balanced toward the acceptance of other family models like adopted child. And those individual benefits has to be balanced to societal risk. And of course, as we said, there are going to be very few individuals who could benefit from opening the door to germline editing at this time. And this could actually potentially open the door to uses which go beyond what was initially conceived to be suitable. Including additional aspects, like I mentioned, pressure towards eliminating rather than supporting disability and fostering, in the long term, inequality, the edited versus unedited individual in a society. So regulation for this potential trial of genome editing at the germline level would probably fall under, again, existing clinical trial regulation, again, human gene therapy providing a framework here. If done with embryo, would of course fall under the current regulation of embryo research and manipulation. Many countries would not, this would be illegal in many countries, including most European countries, although this is an evolving landscape. And our committee did not address the issue whether embryo research or not would be addressed here specifically in terms of genome editing, but would essentially leave this as an area which can be addressed separately. Now, finally, the recommendation of the committee at this level was that caution is obviously needed and paramount, but caution doesn't mean necessarily prohibition. Heritable genome editing may eventually be permitted based on the ethical rationale of allowing to provide some benefit to those specific individuals who would not have an alternative. But we would realize that there's need for more research to meet safety standard, and those, if eventually being allowed, those trials should be conducted under strict oversight and a restricted set of specific criteria. And this was probably the most relevant aspect, to identify a stringent set of criteria which, if not met, would not justify actually to go forward. And I must say that some of the people in the committee also thought that those criteria may never be, in fact, met. And if that were the case, well, the committee would say, well, we would not support then moving forward. So those criteria, some of these are obvious, some may be less, were absence of reasonable alternative, restriction to preventing a serious disease or condition. Obviously, we are leaving out enhancement here. Editing genes that have been, only genes that have been convincingly associated with a disease or a strong predisposition to that condition. Converting those genes to an already existing or prevalent sequence in the population, so not inventing new genes or new sequences, but essentially reverting those alleles back to a dominant sort of a wild type, you want, sequence. Availability of substantial preclinical and clinical data supporting the risk-benefit assessment. Rigorous oversight during the trial in terms of the effect of the procedure on the health and safety. Comprehensive plan for long-term follow-up, including for the multi-generational. Transparency, consistent with patient privacy. Reassessment of both health and societal benefit with the intention to broaden engagement and input on this aspect. And importantly, maybe most challenging, existing or reliable oversight mechanism that would prevent extension of this application to uses other than preventing a serious disease or condition, essentially dealing with this slippery slope, which is so difficult to deal with. And of course, different culture and attitude may see this as something that you may or may never actually effectively achieve. And finally, as I mentioned several times, the need for public engagement. Of course, these were the proposal and the recommendation from a committee of a restricted number of people, though we tried to open up our work to many inputs, but of course we realized the relevance of broadening that input and continuously increasing engagement, especially where we would have to move towards what we at the moment consider some barriers. For instance, whether we would consider eventually enhancement as a possible strategy for using somatic gene editing, or whether we eventually would decide that its time has come to start the first germline gene editing trial according to the condition that I mentioned. With that, I'll finish acknowledging, of course, the sponsor, the committee members, the beautiful work and the input of many, and this is actually the website where you can find many more actually discussion of that. Thank you very much.
All right, our final speaker today is Alta Charo. She is a professor of law and bioethics at the University of Wisconsin in Madison, Wisconsin, with appointments in both the law school and the medical school there. And she's going to talk to us today about societal opportunities and challenges of genome editing. Alta?
How do I advance the slides? Just using this? Very good. Okay. Good morning, and thank you for the opportunity to speak. I actually found that I was going to change this presentation slightly because in the context of gene therapy in general and germline editing in particular, a lot of the concern that we've heard expressed circles around this notion of so-called designer babies. And our speakers have already emphasized concerns around things such as enhancement that goes beyond prevention of disease, or an undue level of selection that brings us back to an era of eugenics. The point I'd like to bring home during this short presentation is that we need to have these conversations in the context of the evidence of what has happened in the past when similar concerns have been raised around other reproductive technologies. I have to emphasize that the views I express here are my own, and they do not necessarily reflect those of the National Academies Committee that Luigi just presented. So these concerns around both eugenics and designer babies are not new. In the 1970s, artificial insemination raised concerns about eugenics and the prospect that people would look for so-called better sperm. The 1980s was in vitro fertilization and a consistent drumbeat of articles saying that having children conceived in a quote-unquote test tube would lead to children being viewed as something less than human, but rather being treated as products or commodities. In the 1990s and 2000s, we saw the discussion around somatic cell nuclear transfer cloning circle around the idea that trying to replicate a particular genome, particularly if it's your own, represented a kind of unbridled level of selfishness and egotism that again would detract from the humanity of the resulting child. And then more recently, The discussion around mitochondrial replacement also, along with genome editing, raised the concerns around not just selection, but being able to manipulate offspring to the point that you have quote-unquote designers. I should have also, sorry. So what can we learn about what's happened in the, what can we learn from what has happened in the past, and how should that be used as we go forward in developing policies around germline editing? If you go back to artificial insemination from the 1970s, certainly it became a very popular option for couples that had a particular form of male infertility. And we saw a rise in the number of sperm banks, and you can see that each one had various catalogs. Here I've got some examples of some of the catalogs that people use. If you could look really closely, and I apologize, it's hard to see, you'll notice that by and large the characteristics are very closely associated with very basic things, such as overall kinds of appearance. Now, is that because we want to help people design their babies? Yes, but to the extent that they're designing them to resemble their partners. The one area where there was tremendous concern about designers was so-called genius bank sperm bank babies. This is Robert Clark Graham, who was an optometrist who had observed that poor people have more children than rich people in his neighborhood. He thought that was bad. And so Robert Clark Graham decided to start a sperm bank with so-called geniuses. The only Nobel Prize winner who ever had semen deposited was Shockley, and his semen was never used. When I interviewed Clark Graham back in the 1980s, he confessed that actually most of the semen came from graduate students and assistant professors in the math and natural sciences. I asked him why he never used anybody from social sciences and humanities, and he actually said, with the social sciences and humanities, you never really know how smart they are. More importantly, he gave this out for free, so the idea was that somehow women would flock to this particular sperm bank to get superior sperm. The fact is, it never happened. We have just a few dozen children that were ever born after having been conceived using the semen, even though it was free. And I thought perhaps this was because Americans are anti-intellectual, and if there had been a sperm bank of famous athletes, that would have gotten more attention. Well, there was for a little while in New York something called the Viking sperm bank that purportedly had semen only from men from Scandinavia, who were presumably very tall and muscular, and it too did not turn out to be a success. Now, the reason is that rather than designing children in the way that was feared, what we really found, I keep finding I'm in the wrong order. Let me apologize and just say the similar kind of parallel discussion was about egg donation, and would we start looking for idealized egg donation. And here we began to see advertisements on some college newspapers for girls that were supposedly more intelligent because they did well on exams. And the concern was that we were going to go for smarter or better looking, particularly better looking girls. And so you found things like these websites, Ron's Angels, for egg donation, that were going to give you the eggs from models. It turned out, actually, if you clicked through, it was a pornography site. Because, in fact, there was no business that ever successfully advertised itself to sell so-called superior eggs. Now, the reason for this, I'm finally getting to the slide I meant to get to, is that when we do surveys of what people really want in both sperm donation and egg donation, by and large, what they want is a healthy child who resembles their partner. Now, of course, if you're single, that doesn't apply as far as the partner goes, but for couples, it's really about creating a family that does not immediately signal to the public that there's something other than the most common form of conception through sexual intercourse, so you don't need to be answering questions all the time. The only areas where you actually began to get a little bit of this kind of eugenic thinking was on height. And I think you can tell just from how I'm standing here that I'm a big supporter of more height. And also the idea of donor's intelligence. I think this is the one area where it's going to be very important over time to help the public understand what is or is not heritable, let alone what is or is not intelligence, because of the widespread misunderstanding of biology in this area. But beyond that, we actually did not see people looking specifically for the kinds of characteristics that other people had imagined in terms of designer babies. Instead, what we had was a desire to match to the non-biological parent, ethnicity, coloring, interests, etc., with basically, as this mother says at the end, the fact that the child looks more like the non-biological parent helps the transition. 1980s, in vitro fertilization. From the time that this first was hitting public attention in the late 1970s all the way through into the 1990s, there was a repeated concern that this was so unnatural that the children would be products, not children, that we would change our way of thinking about them. Indeed, Life magazine actually, and I pulled this out in red, asked, will parents still love their children if they're conceived through in vitro fertilization? But the reality, again, is that IVF did not, in fact, turn children into commodities, and it did not change how parents view their children. And the kind of parent-child relationships that have been studied have not been adversely affected by the form of conception. Of course, those of you here who are thinking about this are going to know this is not a random sample of parents. If anything, it's a highly motivated set of parents because they needed to spend money and go through very uncomfortable medical procedures in order to conceive the child. So in many ways, it seems counterintuitive that they would feel that their children are anything less than an actual gift once they have succeeded through this particular means of conception. What about screening for various kinds of disorders? Again, concern that we were going to become more and more willing to screen for things that are not serious diseases, and instead we're going to be screening for things that are frivolous. This is, again, exactly the same kind of concern that has been discussed since the 1970s, starting with carrier testing, going back actually to the 1960s, amniocentesis, and then chorionic villus sampling in the 1970s and 80s. More recently, and there have been several presentations on non-invasive prenatal testing. And then, of course, pre-implantation diagnosis, looking at embryos, and then only selectively implanting some and not others. Repeatedly, the same concern that we will become less and less tolerant of the kinds of children we're willing to conceive. Question, has that actually happened? Well, take one of the examples that people talk about the most, and that would be Down syndrome. It is true in the United States that about 3/4 of expectant parents who learn they are carrying a fetus who will be a child, who will develop into a child with Down syndrome, will choose to terminate the pregnancy. But that is misleading because the people who are not inclined to terminate the pregnancy often simply refuse to be tested to begin with, because they don't want to know or don't, in their view, need to know. And so if you actually look at the reduction in the numbers of babies with Down syndrome born in the US, from 2006 to 2010, it was 30%. It might be a higher rate of actual desire to avoid Downs because, of course, the age of conception has been changing, and that also changes the underlying rate at which you're conceiving children with this particular triploidy. But nonetheless, the underlying point here is that what we have not seen is a realization of our fear that we are going to use these kinds of tests in order to eliminate certain kinds of people. That said, certainly there are those of us who are worried that the very phenomenon of being able to select against certain disabilities will lead us to become less tolerant of those who have those disabilities, regardless of how it is that they came to have them. But interestingly, and again I speak out of an American perspective, at the very same time that we have watched this steady increase in our control over reproduction, from carrier testing to artificial insemination to IVF to PGD, through all of those things, when we worried that we were going to have fewer and fewer people with disabilities and become less tolerant, we in fact have actually improved the situation of people with disabilities in the United States. Our cities now routinely have buildings and sidewalks and roads that are accessible to those with various forms of physical disability. Employers are now routinely hiring people with Down syndrome and other kinds of cognitive disabilities, and we have more social services available for those people. In basic civil rights, we've seen tremendous advances through the Americans with Disabilities Act that was signed in 1990 by George W.H. Bush, and then further amendments to continue strengthening those protections under George W. Bush in 2008. So on the one hand, we have this fear that each technology will make us less and less tolerant of people with disabilities, but at the same time, if anything, we have becoming more considerate and protective of those with disabilities, at least with regard to our formal lawmaking. So we get to germline and heritable changes, and again, the constant concern about so-called designer babies. Now we're going to use this technology, which Dr. Charpentier has noted is getting easier and easier to use, in order to make changes that are more and more unimportant or are focused on enhancement rather than on cure and prevention. To that extent, we already have, for example, the Oviedo Convention, which specifically prohibits this. And there are a number of countries represented in this room that have implemented this convention through national legislation. And I won't spend any more time on this because Dr. Ormond already noted the range of prohibitions that we have seen around the world. Suffice it to say that a legal prohibition on germline editing exists in many, many countries. And in the United States, although we don't have a formal prohibition on this, we do have a particular limitation on our Food and Drug Administration that prevents it from reviewing any proposal that somebody might make to begin a clinical trial involving an edited embryo. Edited embryos technically fall under the category of human biologics and cell therapies, and so goes to our FDA. So effectively, it is not possible to do this legally in the US. So let me conclude by simply asking, what have we learned, right? Is there really any evidence after 75 years of reproductive technologies that we are really treating our children as commodities, or that we're treating the disabled as something worse than before? And I would argue no. Now, that's not to say these technologies have not had some problems and some abuses, but as a general matter, I do not think personally that these apocalyptic visions of a future filled with children who are viewed as products has ever come to pass or is likely to come to pass by virtue of any one of these technologies. I think the bigger issue is really not those fears, although we talk about them. I think the bigger issue is more fundamental and difficult to describe. It's the question of whether, if a scientist is doing this, it's conveying a message about this. I do believe that this is really at the heart of these discussions, more than any more concrete discussion about the specific consequences of these technologies. There is a consistent kind of discussion around the notion that humans simply should not, quote unquote, play God. And it's a vision of a God that is directive and that takes a personal interest and a hand in directing what happens to each person, and therefore it is not for us to make those decisions. But I'd point out to you that that is not the only way to view playing God. And for those of you who are interested, I highly recommend this novel, Mendel's Dwarf, in which an achondroplastic dwarf is actually faced with the opportunity to choose among embryos, some of which have this dominant Mendelian trait, and some which would develop into children with average height. And he asks himself, am I playing God if I choose? And then he remembers that nature does this by statistics. Nature does this randomly. And from his point of view, to actually choose is not to play God, but to play human. It's an entirely different way of looking at the phrase, and one that's worth debating as we see how much this is influencing our debates. Joseph Fletcher said that man is a maker and a selector and a designer, and the more rationally contrived and deliberate anything is, the more human it is. The real difference is between accidental reproduction and rationally chosen reproduction. So, the report that Luigi Naldini talked to you about was our report. It came out on Valentine's Day. I like to talk about the Valentine's Day massacre of genetic exceptionalism because we said that crossing the germline is no longer unthinkable. And one of the members of our committee had, I think, the best quote in the newspapers: "It's not about designer babies, it's about healthy babies." Thank you.
Thank you very, very much to all 4 participant speakers. So now we open the floor for discussion, and you are all invited to come forward to the microphone and ask questions to our panel. And we have about half an hour for that, so time enough to come forward, introduce yourself, and ask a question. And I would actually specifically like to ask people from the European Society of Human Genetics who have actually, in the beginning of this year, published their own statement and are not yet on stage here, but of course are well represented. And I therefore give first the floor to the lady on the right. Yes.
Martina Cornell, indeed from the European Society of Human Genetics, and I collaborated on those stances of ESHG. In several of the talks we mentioned alternatives like PGD, embryo selection, and if there are alternatives then this would not be a road to go, so we wouldn't choose germline gene editing. But when you think about the ethical consequences, isn't discarding embryos worse than repairing embryos? So that's to the two people in the middle.
Our committee actually addressed that. I'm going to let Luigi follow up with this. The notion of alternatives really focused on whether there were options for somatic therapies following birth that would be just as effective and would not entail some of the risks that germline would entail. But you're quite correct. For some people, embryo selection or abortion are unacceptable and would not be considered alternatives. And they would, in fact, prefer to edit an embryo in order to save it.
Yeah, I don't have anything else to add to that.
And when you think about alternatives, quite a high proportion of people don't get pregnant after PGD, so the success rate per cycle isn't that high. So that's why some of the also parents' organizations are now saying we should really rethink this difference, and in ethical terms it's well, maybe better to repair an embryo as you just mentioned it.
Any response to this?
No, I think it's, I fully agree. I guess the issue is, of course, as mentioned, currently there are technical aspects that would prevent, essentially, to reach the risk-benefit level to bring those considerations in. But once we establish the safety of the procedure, we should consider those aspects. I would agree.
Thank you.
Okay. Yeah, question here in front.
Shukrat Mitalipov, and I'm from Oregon Health Science University, and I specifically do research, at least in vitro, with human germline gene correction. We actually don't use the word of modification or even editing because we're trying to correct mutant genes back to normal. So I had actually the same comment regarding the set of criteria that must be met in order for us, for example, to move from in vitro work to responsible clinical trials. I think I agree with most of the points you raised, but I think that number one that has been brought about in the lack of other reasonable alternatives. So many committees actually would not understand it, for example, at least mine in Oregon, so they think that it shouldn't be any alternative for PGD, and meaning that, for example, for us to enroll in a clinical trial, parents, we have to have homozygous mutants, both parents, for example, if recessive disease, which never exist in reality, because most of the homozygous parents wouldn't survive to adulthood, let alone to be married, to find each other, and willing to have another child. And for dominant mutations, also if there's one parent has to be homozygous mutant, and actually I've been searching for 2 years for any of these, the surviving adult willing to donate sperm, and we actually could never find. So that means we would never meet this criteria if you wouldn't, for example, explain to society that what you mean. So I think your explanation was that maybe some patients would not accept the selection, PGD selection as alternative. So I think if the committee would make it more clearly what that means by no alternative, because that seems like a sticking point for at least in some practical aspects of moving forward to clinical trials.
No, I agree that currently the alternatives are the one that we mentioned, and there are limited cases where you would find essentially a situation where there is no alternative. Although these may be growing, for instance, double homozygotes for disease who are becoming more treatable, they may reach now, rather than in the past, the age in which they may bear children, and they may it's possible that these people are essentially socially pressured into meeting people with the same type of disease, and so there may be situations where couples actually are both homozygous, not carried but affected individual who may decide to have a genetically related child. So this may happen more and more.
Yeah, the most likely these type of families probably would never agree to be enrolled in these clinical trials if they willingly want to have a child with the same condition. So if we say, oh, let's enroll in this, so they have to be willingly enrolled. So we may never actually find any family that would be eligible for this treatment.
But that's maybe an important aspect, because and then if you put in this issue of balancing individual versus societal, aspect, I mean, really opening the door to this challenge, new technology, if there are no firm foundation for doing that, it's a good way of approaching that. So essentially, it has to come from the urgency and the existing need, which is unmet, clearly, to really bring some, as you have heard, there is a substantial concern yet at that level. So I think at the moment, this is, to me, remains a key aspect. I mean, although there are ethical aspects, we agree, but they have to be balanced at the moment. So.
Thank you.
Okay. I think every family is going to have a different sense of how that risk-benefit weighs out as well. And as you said yesterday in your talk, so many of these families, when they don't have embryos that they can otherwise implant, they may be willing to take that risk. And I think that's what we'll need to sort of consider as we move forward.
Okay. Yes, Jean-Louis.
Yeah, Jean-Louis Ondel, Strasbourg. One thing about a possible and not so rare condition, it's a couple, they are both homozygous for APOE4, which is actually pretty penetrant form of late onset Alzheimer. So I wonder whether that would fit in what Luigi Naldini told about, conditions.
This was mentioned, I think, also in our discussion. It's, we mentioned prevention of disease as well, or disease associated. So I think depending on the level of scientific confidence of that aspect, it's debatable, like I say. We cannot give you a recipe. So I think we consider that as one of the firmest evidence of a risk factor which was strong enough to potentially justify consideration for that. That's the only thing I can tell you. The other thing I want to ask also, this is more really should comment on this reproductive biologies. But there is also an issue about the number of embryo that can be generated for PGD, which may be very limited in many cases. So when we say alternative, it may also has to be grounded in realistic alternatives. So where in fact, so there may be a situation where the issue is coming from the fact that there is really no possibility of selecting those embryos because there is a paucity of them. So it may become, eventually this could be the condition where you may consider an alternative, as I know, following the discussion before.
If I may just add just a few words of context too. So this report reflects work that began in December of 2015 and concluded in February of 2017. Throughout that time, there was a lot of discussion around off-target effects or failures to actually achieve the kind of efficacy that you hope for. And given those kinds of uncertainties, there was a real discussion around the kind of caution that one must have when you're making a change that could reverberate down the generations. So both the fact that the people affected could not themselves consent and the potential multiplication of effects led to the conclusion that one should be somewhat conservative without calling for a prohibition and ask that people look at other possible ways they can satisfy their desire to have a family before taking what might be a step that has the most uncertainties associated with it. It was our way of trying to be cautious and not close the door completely. And of course, with every year we learn more about the degree to which those risks are in fact significant, and that will change this discussion over time.
I'll add also that on the point of what diseases might be considered appropriate, we've certainly had these discussions around prenatal testing and PGD for adult onset conditions or conditions with lower penetrance. So I don't think we need to reinvent the wheel there either. We can really look at the literature that's out there and the experiences of individuals going through that.
All right, I see a lot of people at the microphone, so we'll go very quickly with short questions and short answers from the panel. In the back. Yes.
Hi, Jane Tiller from Australia. Thanks so much for some really interesting talks. My question's directed at the final speaker and on the point of whether we are intending to or in fact successful at avoiding certain conditions. And you spoke about Down syndrome and you gave some examples from the US, but I'm interested in your comments on, in the news last year we saw a lot about Iceland and then some other European countries that are actually talking about virtually eliminating Down syndrome through termination. And as a corollary to that, my understanding is that in the US, some of the reason for the figures is the fact that there is difficulty with accessing abortion in the US and that some people say it would be significantly higher if abortion laws were changed.
Well, I don't have enough data to answer the last point. It's certainly a possibility. That is, that the difficulty geographically and logistically of obtaining an abortion affects whether or not people can terminate for that reason. As for European countries, what we've seen is real variation around the world in terms of tolerance for this particular disability. And what is not clear to me is whether or not in some of those surveys in those countries, we are really collecting information about the people who are simply avoiding any decision to be screened at all. Because that really is the first big decision. Beyond that, I don't have the data. When I looked for information on Downs, I found it mostly in the US, a little bit in Australia actually, but those were the only countries, and England with non-invasive prenatal testing. Those were the three I found that I was able to read.
All right, question on the left, yes.
Francesca Forzano from London, UK, and also a contributor to the European Society of Human Genetics papers on genome editing. So I would like to go back to the safety issue, particularly when we might consider that it will be safe enough. Our current framework actually evaluates safety as side effects in terms of quantity, so frequency, but also quality. So we evaluate in vitro, in vivo, which kind of side effects we might expect from whichever kind of treatment. In the case of genome editing, the side effects are relatively random on the basis of where this off-site event will take place. So the question is, do we need a different framework to evaluate the safety in this respect? And if yes, will it be reasonable from a healthcare perspective? Will it be ethical? And in terms of the phase 4 evaluation, I mean the long-term follow-ups of side effects and safety, we will basically turn all these individual in research objects. And will this be acceptable from an ethical point of view, an individual point of view?
Okay, that's a very long question. I would ask Emmanuelle Charpentier to briefly say something about the safety, and then perhaps one short other person responding.
About the safety of the technology. So if you talk about off-target effects, indeed this has been going on as a debate. I would argue that if it's for treatment of diseases combining gene and cell therapy, there is the possibility to really verify that the cells that would have been engineered with CRISPR-Cas9 would have been engineered in a correct fashion prior to reimplantation into the patient. But surely this is what I said. This is why I believe that actually it is important to carry on the debates with regard to direct intervention of CRISPR-Cas9 for the treatment of human diseases or in the human germlines. But the technology is quite young and has also to undergo further development with regard to the safety. What I mean by this is that if CRISPR-Cas9 is used directly, to not have off-site direct effects. If this answers the question, or...
Yeah. I think we should move on because we have about 6 speakers left. So, Guido, yes.
Thank you. Guido de Wert, also a member of the PPPC of this society. My question is linked with both the question of my colleague, but also linked with Martina Cornell's question and linked with a few points that you've made. It's about the relation between embryo selection and modification or editing in the embryo, the zygote, or the precursor germ cells. Mostly, we think, and people, commentators argue, that either you could opt for, say, traditional types of selective reproduction, like PGD particularly, or maybe in the future, the repairing. And then the suggestion is then as if we should think about these options as alternatives. The point that we've made in the European document is that there may well be some sort of smart combinations of the two, both in fundamental preclinical research. When it is about safety studies, one might think of making use of comprehensive genome-wide embryo screening to identify possible off-target effects in the context of preclinical safety studies, but also when we, somewhere in the future, would introduce clinical, that is to say, reproductive germline gene editing, we might think of adding comprehensive embryo screening to check edited embryos for possible off-target effects. I'd like to have your comments. If this holds true, then the two are not alternatives, but might be combined and maybe should be combined. Please.
Okay, short answer from one of the panelists, please.
No, absolutely.
Perfect.
I mean, again, I think that the point about the word alternatives in that particular report that we wrote was simply to say that for some people, there are going to be certain actions that will be unacceptable. And we actually did talk about the dilemma of people who find the discarding of an embryo unacceptable, but yet want to modify an embryo, and our need to check to make sure the embryo is not going to result in a child with some terrible set of problems. And it's one of the reasons why we thought germline editing was really not ready yet, because we didn't have enough information, and the research hadn't been done yet in order to figure out how much we need those things.
Okay, next one. Okay. Sorry, we have to move on. Diane Donnell.
Hello, I'm Mona Miller with the American...
Sorry.
Oh,
in
sorry.
front.
With the American Society of Human Genetics.
All right.
And I wanted to point to Dr. Charo's remarks. Curious, your final statement, the slide said it's not about designer babies, but about healthy babies. But your presentation also highlighted the role of the media in shaping the narrative towards the worries and the fears. And I wonder if you have perspective on social science research by smart social scientists about how the role the scientific community can play in shaping that narrative, and what we all can do to help shift the conversation towards the benefits and the health applications, and balance those fears or worries.
Same thing for you. I mean, in some ways, it's the same thing that Kelly was talking about, so I might turn the microphone to her.
Yeah, I feel like one of the things that we need to really try to avoid is the sensationalist language and falling into the trap of repeating back some of those terms and trying to craft some messages that we can all share a bit more uniformly that embrace the concepts you're talking about, Mona.
All right, yes.
Diane Donnell from Manchester in the UK. Given the huge number of people that can potentially benefit from somatics gene editing, do you think laboring the debate on germline genome editing and having all these reports, et cetera, when you know most people in the public won't be able to separate in their heads genome, germline editing from somatic editing? I mean, I just think that there are so many people that can benefit from this technology, from the cell technology, that in some ways would there not be advantage in just putting this to bed or putting this to one side for some time, concentrating the efforts of public engagement on the potential benefits of somatic therapy, see how that goes, and then see whether in a smaller number of people one needs to come back to this?
Oh, absolutely. Thank you for raising that point. We fully agree with it. I think, in fact, that was one of the key goals to make sure that there was not, the new technology would not bring fear or exaggerate response, preventing what currently are conceived the most obvious and safest applications. So there was a strong support for somatic gene editing in the report. This continues to be, I think, for most of the discussion throughout the society, for responsible testing along what gene therapy is doing. And that will be, of course, provide information to assess the safety. Somebody mentioned random they are not randomly off-target event in this technology. You can predict that, and we are learning to do this, so we'll be better and better able to address the actual risk. So it's not going to be a general genotoxic approach. It's going to be something which can be assessed more carefully. So I fully agree that we should not miss that focus.
Okay. In the back.
Roxandra Bachmann from Zurich. I have a bit of a more technical question. So the main reason I can see from today why we even discuss germline editing over PGD is the instances where there are a paucity of embryos or where people don't want to discard embryos. So today, how efficient is gene editing in situations where you have only few embryos, and how many would you discard because of missed editing?
Emmanuelle, you want to answer this?
Yes, I'm not sure I'm qualified to answer the question because my research is not really focusing on the aspects of safety of the technology for human embryos. Maybe some in the audience know the latest publications in this regard. And
I think you're right. In embryos, this is very inefficient yet. So that especially if you are to edit back, to repair to a new sequence, this is very inefficient. I fully agree. So that's an issue that has to be addressed technically, but it's doable.
Okay, good point. We need to move on. Yes, here in the front.
Hello, I'm Alexander from Republic of Moldova, and I have some questions about ethical side of this problem.
One question.
One question. Yes. Okay. Why should we support dying or handicapped persons and not eliminate their disabilities if we can spend once a bit of resources in a kind of national program to allow access for this technology for all the society, and remove these disabilities from society, remove their impact on society, make lives of people better and longer?
I think what you're asking is why should we not just try to eliminate all of these impairments and disabilities? Is that correct? If we could do that in an equitable way? We couldn't quite hear.
I think
that was the question. Okay. So I think that in concept, there are many ways to agree with that. And then when you talk with individuals who themselves have these disabling conditions, we will sometimes hear a lot of information about how this condition is truly a part of their identity and not just something that they feel they would want to eliminate. And in fact, in the study that we had some qualitative data from, individuals with blindness really reflected on how sometimes this was the best thing that had happened to them, even if they acquired it later in life. So I think we don't want to make a uniform judgment that eliminating all impairments and disabilities is the right thing to do, and there's going to be some nuance to that.
All right. Thank you very much. We go to the question there.
Frances Flinter from London. Germline genome editing is already happening, and there are a number of babies who've been born around the world following mitochondrial replacement therapy. For women who carry a high load of abnormal DNA, PGD may not be an option for them because most of their embryos will have unacceptably high levels of abnormal DNA. In some countries, mitochondrial replacement therapy is happening in a very highly regulated environment, following the sorts of excellent guidelines that you've presented. But in other countries, it's happening in a completely unregulated way and for reasons such as unexplained recurrent IVF failure. My question is, what can the international community do to ensure that the sorts of excellent regulations that you've described are applied throughout the world and not just in countries that accept the benefits of strict regulation?
I think this is a perfect final question, actually. So.
Yeah, I mean, I think in some ways you may be referring to some of the recent work out of the Ukraine, which is highly unregulated, versus what's going on in the UK, where specific licenses are given for specific criteria. And you're exactly right. The answer is we can't. There is no international planetary governance with enforcement power on this point. And so it is through example and persuasion only within the scientific and clinical communities, with a kind of concept of professionalism, and also over time by demonstrating the success of places like the UK at making this available in a responsible way to persuade governments in other countries to adopt a similar approach. But there is no way to ensure it. And medical tourism will continue. If people find that they are unable to be satisfied where they are, they will find it someplace else.
This is a perfect ending of this debate. I want to thank all the panelists, and I want to thank you for attending. Thank you.